Okay, hey Biochem. So today is first day of electron transport chain, which is abbreviated as ETC. There's quite a bit of detail today, so pace yourself, brace yourself. After watching this video, you should be able to calculate the delta G from reduction potentials and combining half reactions for each complex in the ETC and the overall process. You should describe the flow of electrons through the ETC, state the eukaryotic cell location of the ETC, describe the purpose, state the mobile electron carriers and how many electrons they can each carry, and then state the other electron carriers, within each complex. So there's mobile electron carriers, which means they move, and then there's these other electron carriers, which are fairly stable. So before we get into the ETC, I kind of want to give you a big picture of what we're doing so that maybe you can come back to it if you feel like you're getting lost in the weeds of the details. All right, so what we've done so far is we've taken glucose, which has 6 carbons, and we've oxidized it to 6 CO2s, and that went from glucose going into glycolysis, and then to PDC, and then to Tca cycle. And we've also taken a fatty acid, another food that you eat, and oxidized that to CO2. And let's say it was a 16:0 fatty acid, then we should get 16 CO2s out of it because CO2 only has 1 carbon, so you have to make sure that you get the complete mass transfer. All right, so we've done that part of the combustion reaction. The part that we haven't done yet is when you oxidize something, another something has to be reduced. So in our cells, our human aerobic cells, all these electrons are passed to a water, which is then reduced to, these electrons are passed to an oxygen, which is then reduced to a water. So you notice that the oxygen is gaining electrons, which are in the form of hydrogen, so the oxygen, the O2 is getting reduced, while as the fatty acid is getting oxidized. And instead of being on fire, having little flames going on inside of every single cell, like we would normally do if you just put a lump of fat on the table and lit it on fire, what has happened is while oxidizing glucose to CO2, you've been moving those electrons to these reduced electron carriers called NADH and FADH2. There's no particular reason that I wrote these in opposite order. Just FADH2 was on my brain first. So these come off of pathways like the Tca cycle, glycolysis, PDC, beta oxidation. Basically every pathway and cycle that we've encountered so far has given off an NADH, and then a couple have also given off FADH2. So the electrons from glucose are going to be passed to oxygen, and then these will be regenerated. Okay, so we're at the point where we can start talking about reduction potentials. But maybe first, before we get there, maybe we're going to talk a little bit about why is it that the electrons get passed to oxygen? So, or maybe the question is, why do we have to breathe O2? There are some anaerobic organisms that don't need oxygen. They can use other things to accept their electrons or electron transport chain. But for us, why is it O2? Why are all the electrons going to eventually end up on an oxygen? And it turns out that oxygen has a positive reduction potential. Which just means that it wants electrons really bad. Really, really badly. It wants electrons a lot. because when you pass electrons to something with a positive reduction potential, it ends up being a very large negative delta G, which is favorable. And we're going to do the math to prove that to you. Okay, so here are two half reactions. There's one right here and one right here, and their reduction potentials are on this side. And you'll notice that the reduction potentials are always written in a way where the electrons are on the left side of the arrow. Now, if we have something that is reduced, something must be oxidized. So when you combine these half reactions, one of them has to be flipped. So I want you to use a little bit of like what you know about what's going on to figure out which one's getting flipped. So I know that we're going to pass electrons to oxygen, and that oxygen is going to become water. So that equation is already set up in the correct order, which means that this must be the equation that has to get flipped. So in this case, we're going to flip the top one. So it's going to be NADH goes to NAD plus a proton plus 2 electrons. And because I flipped the half reaction, you're going to flip the sign on the reduction potential. So this will be a positive 0.320 volts. I get to leave my second half reaction the way that it is. So half of an oxygen plus 2 protons plus 2 electrons goes to water. And because I didn't change that half reaction, nothing changes here. So plus 0.8166. And before we do the math and before we add these, you should notice that this half reaction has a positive reduction potential, which means that it wants electrons a lot more than this one right here, which has a negative reduction potential. Okay, so we're going to add these half reactions together. Before I do that, I'm going to cancel out anything that's the same on both sides. So those 2 electrons cancel out those 2 electrons. They're on opposite sides of the equation. I've got 2 protons here and 1 proton there, so I'm going to actually just cut that one off and cut that one out. And then I think everything else looks okay. So if I add those 2 half reactions together, I get NADH plus a half of an oxygen plus a proton goes to NAD plus H2O. Now I just want to make sure that it looks balanced. I've got 2 hydrogens here, 2 hydrogens there, an NAD, and NAD. A half of an O2, which means there's only technically one oxygen there, and 1 oxygen there. And then just in the same way that I added the reactions together, you also add the reduction potential together. So this is going to be delta E, and when I add those 2 up, I get 1.1366 volts. And to calculate the energy of moving those electrons from NADH to oxygen, I'll put it into this equation that you'll be given. So delta G equals negative N is the number of electrons. The negative in front of the N is accounting for the sign on an electron, so that it's negative. So I'm going to put 2 in here because I had moved 2 electrons. 2 electrons, you don't add the electrons up together, it's just 2 electrons moved from NADH to oxygen. F is Faraday's, 96500 joules, mole, volt. And delta E is what you calculated up here, 1.1366 volts. So even before you do this, you should see that as long as your reduction potential is positive, this will be positive, a number of electrons has to be positive. The overall value of your delta G will be negative. So as long as your reduction potential is positive, then the electrons are moving in a favorable direction. So the delta G of this comes out to be negative 219,363. I'll add some extra numbers here. Joules per mole. The reason it's joules per mole is because that volt cancels with that volt. Electrons are unitless. That's a lot of energy. That's a lot. what if I asked you how many ATPs we could make from this? So how many ATPs can be made by passing electrons from NADH to O2? Well, you'd have, this is a delta-G of, I'm going to put O2 reduction. That's this, plus the delta-G of ATP synthesis, comma S. And we're going to max it out, so we're going to set it equal to 0. And I want to know how many, so I'm going to want to figure out that coefficient right there. And so you'll plug in what you have, excuse me, my poor voice. Let's see, negative 219,363.8 joules per mole plus the amount of ATP, the number of ATPs. And I know ATP synthesis costs me 50,000 joules per mole per ATP. Set that equal to 0, do your math, and you get something that is like 4.4, is that about right? 4.4 ATPs, which you can't have 0.4 of an ATP, so you need to round down because, again, we cannot create mass here. So that's about 4 ATP per NADH. That's a lot. That's per just one NADH. Now, I think I've told you in class that an NADH is actually only worth about 2.5 ATPs, and we will talk about that when we get to ATP synthesis, like why it is that we're losing some of this energy. And really it just has to do with the fact that the whole thing is just not very efficient. Like we're just going to lose little bits of energy as we go along, so we don't capture all the energy. But if we were to capture all of the energy, every NADH should be worth 4 ATPs. Again, it's not going to be worth that much, but just based on this math, it should be worth 4 ATPs. Okay. So that's the big picture. So the big picture is we're taking electrons from NADH and we're putting them on an oxygen to create water. The oxygen is what you breathe in. The NADH comes from pulling electrons off of the food that you eat. As you pull electrons off the food that you eat, glucose and glucose and fatty acids, they become CO2, and you exhale out that CO2. Okay, so here's a diagram that I just pre-drew because I've been drawing it almost every single time, and that must mean that it's important, but it's to help you see the connection between things. So I've pre-drawn this out just to save a little bit of time, where we have glucose catabolism here, where a glucose enters the cell, it gets converted to pyruvate. The pyruvate enters the mitochondrial, this is the mitochondrial matrix. On the inside here, I've just drawn the mitochondria as a circle. The pyruvate is converted into acetyl-CoA plus a CO2. The acetyl-CoA goes into the Tca cycle and spits out more CO2s. And then along the process, here in glycolysis, we get a little bit of ATP out. We get a little bit of NADH. Some of the pyruvate is converted into lactic acid in fermentation, which spits out an NAD+, which is used to regenerate, to keep glycolysis going. Over here, with the bridge step, we get out some NADH. We also get out NADH of the Tca cycle, 3 of them per turn, FADH2, and GTP. And then these carbon dioxides, the final product of glucose metabolism, are exhaled out. Okay, so the CO2 that you breathe out is coming from the food that you eat. All right, so if we follow the path of a TAG, or a fatty acid, a TAG goes in, it's converted into glycerol and a fatty acid. Those carbons from glycerol are not wasted. They enter in kind of halfway through glycolysis, not quite halfway through, but about. The fatty acid carbons are first converted to acyl-CoA, which is then converted to acyl-carnitine for transport, which is then reconverted back to acyl-CoA, and then that goes around and around and around again, chopped off 2 carbons at a time into acetyl or acetyl-CoA, and those 2 carbons that are coming off every time are finally oxidized again by entering into the Tca cycle and coming out as CO2. Beta oxidation also spits out 1 NADH and 1 FADH2 per round for a saturated, for a fully saturated carbon acyl chain. Okay, so that is a very simplified version of what we've done so far. What we're going to do now is we're going to take all of these electrons and we're going to get some energy out of them. Okay, and it's no coincidence that most of these electron carriers are inside the mitochondrial matrix, because where we're going next, and let's see if can I rotate this? No, but I can zoom. Where we're going next is we're going to the inner mitochondrial membrane, which is where the electron transport chain is, and we're going to put 4 complexes. These are all proteins, they're all enzymes. We're going to put 4 complexes on the inner mitochondrial membrane, and we're going to go through the electron transport chain. But essentially, all of your NADH is going to go into complex 1, and your FADH2s are going to go into complex 2, and then we'll talk a little bit about complex 3 and complex 4. This is 1, this is 2, 3, and 4 Roman numerals, I think. Here, we'll go here. Who am I missing? Oh, hey, there you are. And then eventually we'll talk about, see this sad NADH that's not on the right side of the membrane? It's like on the wrong side of the tracks. Eventually we will talk about how to get those electrons to transfer over. Okay, so what I'm going to do first is I'm going to draw I'm going to go to a new page and I'm going to draw this zoomed out and rotate it a little bit. So I'm going to rotate it so it's horizontal. Nope. Vertical. Nope. Horizontal. I had it right the first time. Horizontal like the horizon. It's vertical right now. So I'm going to rotate it. I suggest that if you're following along that you get a piece of paper and you turn it the hot dog way, the long way. Basically rotate it 90 degrees from the way that you normally write on it. And we're going to draw, and then we're going to go through a few of the slides that I normally create. We're just going to go through them. I'm not going to draw the words on them. I'm just going to point some things out so that you can have a visual representation of what is going on here. Okay, so here's my new piece of paper. So I'm going to make this the inner membrane, and I'm going to make it really big because I have a lot to draw on here. So maybe that's not even big enough. Maybe I'll make it like humongous. So this is the inner membrane, and it's a lipid bilayer. That's why I have it drawn as 2 lines. And then I'm going to put the outer mitochondrial membrane up here, and I'm just going to kind of shade it in a little bit. Okay, this is the outer mitochondrial membrane, which means that this space in between is the IMS or intermembrane space. And then what's down here underneath is the matrix. Okay, so I've taken it and I've rotated a bit. Okay, so I'm going to draw the complexes in order going from left to right. And I'm going to make them really big because we have a lot to draw in them. So this is going to be complex 1. It's an enzyme. They're all enzymes. They're all giant protein complexes. Complex 2, I'm actually only going to have it going about halfway into the membrane, and there's a reason for this. It actually does span the whole membrane, but there's a reason that I'm going to do this, and I hope that it helps you understand some of the ideas. This is going to be complex 3, and then this is going to be complex 4. 2, 2, 3, and 4. Okay, so let me see, make sure I have the notes ready. All right, here we go. All right, so we're going to start at complex 1. So complex 1 is where NADH, all the NADHs that we have, finally get oxidized to NAD+. And if you remember, this NADH is a 2-electron carrier. And these 2 electrons are going to be passed to a molecule that has a higher reduction potential than NADH, because this whole thing has to move in a favorable direction. So the first place that it goes is going to go to this electron carrier that's called FMN, or flavin mononucleotide. And it's going to pick up those 2 electrons, and it's going to become FMNH2. So it picked up the 2 electrons from NADH because FMN has a higher reduction potential. And FMN is part of complex 1. And then FMNH2 will be regenerated, and in the process, an iron-sulfur cluster, these are pretty cool, will pick up the 2 electrons. It will be an iron-sulfur cluster. I'll put an H2 here, okay, to show that, again, we're passing 2 electrons along the line. And the iron-sulfur cluster has a higher reduction potential than FMN. So, again, we're passing in a favorable direction. Now, the electrons are going to be passed to our first mobile electron carrier, called Q, and this Q is going to come and grab those electrons to become QH2, and we'll talk about what Q stands for and what it means. There's many names for it. But it gets transported to Q, and then this gets regenerated. Okay? So what you'll notice is that for complex 1, the overall is that we start with NADH, and we pass to a Q. Everything else has been regenerated. That's why I've drawn them as these kind of goofy looking arrows, so that you can see the FMN gets regenerated and the iron-sulfur cluster gets regenerated, and we pass to Q, which becomes QH2. Now QH2 is mobile. It can move, and it's soluble in the inner membrane, which tells you a little bit about its polarity. It must be pretty nonpolar to be able to move freely through those nonpolar lipid, phospholipid tails. Now the QH2 is going to go to complex 3, and it's going to drop off electrons, regenerating Q. Now we're going to drop off electrons 1 electron at a time. It will drop off its 2, but it's just going to it's going to only do it 1 at a time. And these electrons are going to pass through a series of cytochromes. So the cytochromes that are going to be passed to, we're going to first pass to, I'm going to have to make this bigger. Okay, so we're going to first pass to Cytochrome B, and cytochromes they have hemes in them, so they have irons in them. So this is going to be an Fe3 plus cytochrome, and once it grabs the electrons 1 at a time, it will be an Fe2 plus. And this is Cytochrome B. And again, I'll show you the structures of these. And then from here, we're going to pass again. So we're going to regenerate here. And a Cytochrome C1. Again, cytochromes have hemes in them. So this is an Fe3 plus. It's going to take 1 electron and become an Fe2 plus. Cytochrome C1. There's a reason we're talking about cytochrome C1. And again, these are, I'm going to clump these together because the iron is the iron in the heme, which is part of the cytochrome. Okay, so we're passing 1 electron at a time. And then finally, the cytochrome C1 is going to pass to a cytochrome C, just a regular C, again, Fe3 plus, to become cytochrome C, Fe2 plus. And this is a protein that is soluble This is a protein that's soluble in the intermembrane space, and this is the second mobile electron carrier. There's an I in there. Come on, man. Electron carrier. So cytochrome C is the first mobile electron carrier, and QH2 is the other mobile electron carrier, and they're mobile because they move. All the rest of these electron carriers don't move. So this cytochrome C is going to move, it's going to float over, travel through the intermembrane space, which is a liquidy area, and it's going to end up at complex 4. And in complex 4, we're going to meet 2 more cytochromes. So there's going to be 5 cytochromes in total. So here we're going to meet cytochrome A, and again, it's going to start out as Fe3+, and it's going to be reduced to Fe2+. So the cytochrome C will come over to complex 4. We'll come over to complex 4, pass its electrons, and then be regenerated into Fe3+. So again, we're only doing 1 electron at a time. Eventually, all the electrons will move over. All 2 electrons will move over. Okay, and then finally, these electrons are going to move onto cytochrome A3. This is the really important cytochrome to remember. So cytochrome A3 will be reduced to an Fe2+. Again, all cytochromes have hemes in them. And then I'm going to zoom back out. Now here is where cytochrome 3 is where oxygen gets reduced. So it's going to grab those electrons and some protons from the matrix, gets reduced to water, and then this is regenerated back. Okay, we're not done. We're maybe like halfway through this story. But that is the flow of electrons from an NADH landing on an oxygen and reducing the oxygen to water. Every new electron acceptor, we go from FMN to iron sulfur to Q to cytochrome B to cytochrome C1 to cytochrome regular C to cytochrome A to A3 to O2. Every single one of these has a higher reduction potential than the one before it. So if you think about like a giant relay race, is that every one that you pass the baton to wants it more than the one before it. Okay, complex 2 is a really interesting one. And, okay, this part always makes me nervous because I always feel like everybody's going to be like, oh my gosh, why is this happening to us? Okay, so complex 2 is an enzyme. And it's an enzyme in the Tca cycle. It's the enzyme that takes succinate to fumarate. Okay, so if I were to draw this, I could draw a big circle here, and this would be the Tca cycle. And this would be where acetyl-CoA comes in, up here. Okay, succinate to fumarate is the step where FADH2 is generated, and it turns out that the 2 electrons from succinate, so succinate is oxidized to fumarate, while FAD is reduced to FADH2. It turns out that this FADH2 is actually part of this, the box that I've drawn here, part of the enzyme. It's inside of it. And the electrons from FADH2 will also pass to an iron-sulfur cluster. Crazy, huh? Again, here we're still passing 2 electrons. And these electrons will pass to another Q. So I'll just kind of swoop it up like this. And this QH2 also drops electrons off at complex 3, one at a time, and regenerates back to Q. So what you should know is that there's just a concentration of Q and QH2 in the inner membrane. And both complex 2 and complex 1 deliver electrons to Q to reduce it to become QH2. And QH2 always goes to complex 3. Okay, so every single complex, the passing of the electrons is favorable. So each complex Okay, so if we were to calculate the delta G of complex 1, I'll just do C1. The delta G of complex 1, where NADH eventually passes just to a Q, would be negative. Now, that energy has to go somewhere, and if you'll notice in the electron transport chain, we're actually not making any ATP whatsoever. So that energy has to go somewhere, and it's going to moving protons. So do you remember our delta G of transport, which was RT ln C2 C1 plus Z F delta psi? Good, I'm so glad. Why do I ask questions that I can never have the answer to? So we're going to actually move protons. So the energy from moving these electrons in complex 1 is going to be used to push protons. So the protons, what color haven't I really used here? Looks like maybe green. Okay, so in the intermembrane space, there's a high concentration of protons. Okay, and so in complex 1, because we're releasing energy, that energy is able to move these protons against their concentration gradient, against their chemical gradient and their electrical gradient. And complex 1 specifically moves 4 protons. It's a number that you need to know. That's a 4. Specifically moves 4 protons. So why? So it's moving it against its chemical gradient. So against it's going from low concentration to high. And it's also because there's so many protons on that side of the membrane, that side of the membrane is more positively charged because we have more protons on the intermembrane space side. We also get an electrochemical gradient. So we get an electric gradient. So electrochemical gradient is both of them. All right, so I'm just going to say that one more time. So each of the complexes is passing electrons in a favorable direction, which means that complex 1 is releasing energy when it passes from NADH to Q. And that energy is harnessed in order to pump protons. And those protons are moving against their electrochemical gradient, and the energy is supplied by moving the electrons. Okay, so complex 1 will pump 4 protons. Complex 2 does not pump any. That's why I have drawn it only halfway through the membrane, because it doesn't pump any. Complex 3 also pumps protons. I run out a little bit of space here. It again pumps 4 protons. And then complex 4 spans the membrane and also pumps protons, but here it's only 2 protons. So what you'll notice is that you get 4 plus 4 plus 2 is 10 protons being moved per NADH. Okay, and then FADH2, because it starts at 2, it will only pass electrons to 3 and 4. We get 4, 5, 6 protons, poor FADH2. These are going to be important things to remember. Okay, here we go. I'm going to talk through it one more time. So electrons, NADH at complex 1 is oxidized to NAD+. 2 electrons are moved, stripped off of it, and moved onto an FMN to create FMNH2. Those electrons are then passed to an iron-sulfur cluster, which is then reduced to and is then also reduced. Those electrons are finally passed to a Q to be reduced to QH2. QH2 then goes to complex 3 and drops off electrons one at a time. The first place that those electrons go is Cytochrome B. Then they're passed to Cytochrome C1. Then they're passed to the mobile electron carrier, Cytochrome C, which travels through the intermembrane space and drops off electrons on complex 4. The first place they land is on Cytochrome A. And the second place they land is on Cytochrome A3. And then finally, those electrons are passed to an oxygen, which is reduced to water. Okay. For succinate to fumarate, succinate is oxidized to fumarate, and in the process, 2 electrons are passed to an FAD to create FADH2. Those electrons are then moved again to an iron-sulfur cluster, and from the iron-sulfur cluster, we move to the mobile electron carrier Q that gets reduced to QH2, and again drops off electrons one at a time. So in our half reaction, it said that 1 NADH is able to reduce half of an oxygen. Okay, so that means that in order to get an entire oxygen, we need 2 NADHs. So it's going to take 4 electrons to get an entire O2 to be created into 2 waters. It's just food for thought. Okay. All right, so now, I know this is a big giant mess, but now we're just going to go through some slides that I have created. And so you can see some of these structures so that you can maybe have a better idea of what some of this looks like, and we'll talk about the overall reactions for them all. Okay, so this is just a figure that shows that all of the complexes, well, that was actually worse. All the complexes are on the inner membrane of the mitochondria. So when you're saying the location, it's the inner membrane of the mitochondria. So inside of this mitochondria, they have a Tca cycle, which they've called Krebs cycle, and then they've got NADH coming out, FADH2. So the location is the inner membrane of the mitochondria. Okay, so complex 1 is huge. They're all huge, but here's a schematic cartoon diagram of complex 1, and what you're looking at is not a eukaryote. You're looking at a bacteria, so don't just ignore these things, okay? So here you have NADH going, so that for us this would be the matrix, okay? So you have the NADH going to FMN, which is then passed to these iron-sulfur clusters, and there's actually, I think there's 8 of them. There's a whole bunch of iron-sulfur clusters, and then eventually it's passed to Q, and then QH2, and this pumps 4 protons across the membrane, and the reason we're able to push these protons across the membrane is because the flow of electrons here is favorable. So the flow of electrons is a negative delta G, which is able to be harnessed to move these protons, which is a positive delta G, and eventually you'll be able to do the math for that. Now what is FMN? So here's the structure of FMN, just something to be able to recognize. It's called flavin mononucleotide, and it accepts 2 protons and 2 electrons, and that shouldn't be surprising because it's stripping 2 electrons from NADH, so you have to be able to have something that accepts 2 electrons. What vitamin should you take if you're deficient in FMN, which is flavin mononucleotide? Maybe you remember, maybe you don't, but I'm going to jog your memory a little bit. Flavin is a shorthand for riboflavin. So if you're deficient in FMN, it's because you're deficient in the riboflavin, and I believe that is vitamin B2. Okay, so what's this Q thing? Q has many names. Ubiquinone or ubiquinol is sometimes what it's called. So ubiquinone is the oxidized version, ubiquinol is the reduced version. It can sometimes be called Q, which is what I usually call it, or it's sometimes also called CoQ, which stands for cofactor Q or cofactor ubiquinol. It's the mobile electron carrier. Here's its structure, and the reason that I have this up here is because it has this isoprenoid chain on it, and what you'll notice, or maybe what I'll just point out to you, is that, what color am I using? Yellow. There's 10 carbons right here. 10 carbons. So this is a really long carbon-hydrogen chain, which is super nonpolar, which is the reason why it's soluble in the inner membrane. Okay, so it contains a variable number of isoprenoid units, making it soluble in the inner membrane, because inside the inner membrane is where all the phospholipid tails are. And the reason that it can drop off one electron at a time is because it's able to accept single electrons at a time. So because it's able to accept single electrons, it's able to drop off single electrons at a time, but it can carry up to 2 electrons. Okay, that is ubiquinol, CoQ, Q, ubiquinone. I will just call it Q. I just call it Q and QH2 when it's got 2 electrons on it. Okay, so is the passing of electrons from NADH to ubiquinol or Q favorable? So what I have is a table of reduction potentials for the ETC. And I've highlighted where they're coming from. So they're coming from NADH, which has a reduction potential of negative 0.320. And then I look down a little bit and I see ubiquinone. And ubiquinone has a higher reduction potential. So yes, the energy, this says this energy is used to drive what process, but the energy from going from here to here should yield a negative delta G if we were to add, flip. This one needs to be flipped. You would flip that half reaction, that would become a positive. Add those together and you would have a positive value for your delta E and therefore negative for delta G. And this energy is used to push the protons into the inner membrane space. Okay. If you'll notice, look at here. Here's all the cytochromes, and all the way at the bottom is oxygen. So in complex 3, we go from Q, I need a different color here, Q to B to C1 to C to A to A3 to O2, and then you'll notice over here that every single reduction potential is getting more positive in value, greater and greater in value. So the flow of electrons through the ETC overall is a very favorable event. And all of these are used to push protons into the inner membrane space. Okay, so complex 2, like I said, is the one that takes succinate and oxidizes to fumarate. And in the process is where you go from FAD to the iron-sulfur cluster to a QH2. So in this diagram, they have their complex 2 spanning the whole membrane, which it actually does do. It doesn't leapfrog. These Qs don't leapfrog over that. That's just silly. But I always draw mine just as like a half, so you can see it a little bit better. Complex 2 doesn't pump any protons across the membrane. What I have from here is, so this part of the electron transport chain is also where the FADH2 from beta oxidation enters. It enters in like a little bit sideways. It doesn't come in quite through complex 2, but the idea is that we're passing in electrons from an FADH2 also to a Q. So I usually just have you connect your electrons from beta oxidation from the FADH2 into complex 2. All right, so complex 3 has a whole bunch of cytochromes in it, and it transfers, it accepts them only one electron at a time, and then eventually the electrons are passed to the mobile electron carrier Cytochrome C, and this one pumps 4 protons. So here we're just adding more and more and more of the complexes on. So here's complex 1, here's complex 2, oh, it already says it. Here's 1, here's 2, here's 3. This one pumps 4 protons, pumping no protons, pumps 4 protons, and this Cytochrome C is going to go find a complex, complex 4. Okay, so complex 3 has a bunch of cytochromes in it. It has Cytochrome B and it has Cytochrome C. And so I wanted to point out, again, that these are hemes. The hemes are inside of the cytochromes. And we have a couple different flavors of the hemes. So heme C is in C-type cytochromes. This iron protoporphyrin 9 is in B-type. And you notice that they have a little bit different functional groups coming off of the porphyrin. And B-types are associated via hydrophobic residues because these are nonpolar. And C-types are covalently attached to cysteines because they have sulfurs on them. Okay, so this is just my reminder that cytochromes have hemes in them. And the different cytochromes have different hemes because they have different functional groups. And I'm going to just circle this question because this whole class is cumulative. And so I want you to go back and think about, do you remember why the hemes have reversible binding events? Because the iron could accept an electron and not give it up. But it does give it up, and it gives it up for a few reasons. And I'd like you to go back and think about, like, what are the reasons that the iron in the heme is able to accept and deliver electrons. Okay, so is the passing of electrons from Q through the cytochromes in complex 3 favorable? Yes, we kind of already did this. We're going from Q to B to C1 to C, and all these have increasing reduction potentials, and that energy again is used to push protons across the membrane. Cytochrome C, like I said, is a protein with there's the cytochrome, the heme inside of it, and it's a super tiny protein, and it's stabilized by lysines that are lysine side chain stabilized contacts with complex 3 and 4. So that's how it links on to 3, and that's how it links on to 4, and then it's very reversible and goes back and forth, back and forth. Again, this is a mobile. This is the second mobile electron. I can't spell mobile. Electron carrier. And it carries 1 electron at a time. Okay, so that takes us to complex 4, which is the most important of them all, in my opinion. It's humongous. They're all humongous. This one's super humongous. But electrons are delivered by cytochrome C 1 at a time and are passed along the heme groups and then through some coppers. The final electron transfer is to oxygen, which happens at cytochrome A3. So cytochrome C comes along, passes electrons. They're highlighting this because there is, what do you call this? A copper center right here. And then the electrons are passed to A, and then they're passed to A3. And this is where oxygen comes into A3 and leaves as water. And type A hemes have isoprenoid functional groups, so they're associated with the protein through hydrophobic interactions. And again, the passing of electrons from cytochrome C to A3 to oxygen is favorable. Okay, so here's a summary of all of the things of, like, why is this all happening? So we have a lot of electrons that are being passed through complex 1 that go to Q2, to Q to QH2. QH2 drops electrons off at complex 3, which gives electrons to cytochrome C, which drops electrons off at complex 4, and complex 4 is where oxygen will accept those electrons and be reduced to water. In the process, each of these complexes, except for 2, will pump protons because of the energy released from moving electrons. What this causes is both a chemical potential and an electric potential. So the chemical potential is because we have a lot of protons, a high concentration of protons here compared to here. And because the protons are charged, positively charged, we have an electric potential created because this side will have more positively charged atoms than the side underneath. And I'm just going to write some of the full reactions for this so that you can see that overall really it's just going from NADH into oxygen. So bear with me here. Okay, so complex number 1. Electrons start at NADH, which gets oxidized to NAD+, and they end on QH2, which means I have a Q over here. Complex 2, we start with succinate, and that gets oxidized to fumarate. And again, the electrons are passed to a QH2, which means that we have to add the Q. Complex 3, electrons start at a QH2, which means QH2 is oxidized to just regular Q, and is passed to Cytochrome C, which will be an Fe2+, which means I have to add in the Cytochrome C mobile electron carrier Fe3+. Okay, and then at complex 4, Cytochrome C that has the electron, so it's 2+, is going to drop off electrons, so it becomes oxidized to an oxygen, which will be reduced to water. I haven't balanced out all of my electrons here. You can do that on your own. So I just want to say electrons are not balanced. And neither are the hydrogens, neither are the oxygens. Like nothing's quite balanced here. But what I want you to see is that if we start at complex 1, and then go to 3, and then go to 4, because that's where those electrons are, you'll notice that if I go from 1 to complex 3, that Q and that Q will cancel. And this QH2 and that QH2 will cancel. If I go from 3 to 4, this Cytochrome C will cancel with that Cytochrome C. This Cytochrome C will cancel with that Cytochrome C. So when I go from complex 1 to 3, my overall is NADH plus oxygen goes to NAD plus water. And those are the 2 half reactions that we combined at the very beginning of this. And again, the reason I skipped over 2 is because the electrons from NADH in complex 1 never passed to 2. But we could do that as well. So if we start at complex, so if we go from 2 to 4, we go from succinate to fumarate, Q to QH2. This Q cancels with that QH2 cancels with that QH2. The electrons from the Cytochrome C will be passed to 4, so that one will cancel with that one. And again, what you'll see is that overall, you'll have succinate, fumarate, which passes electrons to oxygen, plus water. The other way to think about this is really FADH2 to FAD. Okay, let me see if I have anything else. So there's a lot to take in here. You're going to have a lot of math to do, a lot of organization in your brain. So just take your time with it, go through your notes, redraw it, come talk to me if you need help, and I'll see you in class.